Hello, my name is Joseph, and today I'm going to talk to you about a new method developed in our group called sciCSR. This method is meant to be for predicting and inferring the dynamics of state transition of B cells in single-cell RNA sequencing data. So B cells are a group of cells which produce B cell receptor or antibodies in a secreted form for immune activity. B cells mature using two processes. The first process goes to somatic hypermutation, which act on a variable region of the B cell receptors, where mutations are acquired to optimize the targeting and binding to the target antigen of the B cell receptor. B cell receptor also contains a constant region, which could also change using a process called class switch recombination. So in class switch recombination, we first need a stimulus to switch towards a particular state. For example, in this case, from IgM, which is the first isotype here, to G1. So the stimulus would act on the genome to trigger the transcription of this particular type of mRNA transcripts, which are called sterile transcript in the literature. They are called sterile transcript because this particular type of transcript contains coding information of only the constant region of the B cell receptor, but not the variable region, and therefore wouldn't encode a functional B cell receptor for the activity of B cells. The purpose of the sterile transcription is to allow an enzyme called AID to access these particular areas in the genome and cause mutations. So the resulting DNA repair of the mutations would result in removal of the genomic segment in the middle. So these will be removed from the genome, and the subsequent repair would bring this target isotype to immediately after the variable region. And therefore, if you then transcribe this particular transcript and then translate it, you would end up with a full-length, functional, and productive B cell receptor. So it's a very complicated process, but the take-home message here is, in the transcriptomic data, we should be able to see these two types of mRNA transcripts, and therefore, if we look carefully enough, it encodes signals which allow us to learn about a B cell's current and future states. So this is the hypothesis that we have, which is, can we build methods that extract these signals and build models to infer transitional dynamics between different cell states of B cells? So we have a new method called sciCSR that allows you to do this. It's an R package that implements a lot of implementation that allows you to answer this question. We looked into the raw transcriptomic alignments to distinguish for each molecule there whether we see evidence of the coding region for the variable domain, which allows to distinguish whether each molecule is a productive and sterile transcripts. Once we have this, we then go back to the transcriptomic data, here represented as a network. We map on top the productive and sterile transcript information and score each cell by where they are along the B cell maturation process. So this score allows to improve directionality on the network so that we can then infer transitions based on class recombination information. So here, this is an illustration done on an example data set collected from the tonsil, where we see using class recombination information, we reproduce what is expected in terms of B cell transition from naive to memory cells to activated cells and also to enter the germinal center. So this is done on class recombination, which is a native process of B cells. We could also do this on somatic hypermutation, where we see very consistent trends in terms of the inferred transition between B cell states. So expectedly, these two processes, both of them native to B cell and is integral to the maturation of B cells, expectedly should produce convincing transitions in the data set. So we try to apply the sciCSR method on different types of RNA sequencing data that is publicly available. One of them are these sort of time course studies whereby the immune response of individuals following immune challenge, in this case, the Pfizer SARS-CoV-2 spike protein mRNA vaccine, can be monitored across time point. So data here show, for example, for one donor, in this case, we see a very smooth and stepwise state transition from IgG1 to A1 to G2 across the 3 time points. So we hypothesize that if we apply the method on this first time point, we should be able to predict the distribution of states at the second time point. So these bubble plots here shows the results of the prediction applying on the first time point. The bubbles here correspond to the amount of transitions between each pair of states. This is indicated by the color of the bubbles. And the size of the bubbles are the comparison to against known models where the transitions are randomized. So we can have an idea about the significance of these inferred transitions. So in this case, we reproduce what is seen in the observed distribution, and we can compare the prediction made to the observed distribution at a subsequent time point, in which case we see a very good similarity between these two distributions. We can do the same thing, apply the model on the second time point and predict the distribution on the third time point, in which case, once again, you see a very good similarity between the prediction and the observation subsequently. So I think in general, this method works quite well in predicting the directionality of state transition in B-cell data. We think that this is because it extracts signal directly relevant to the maturation process, which is the underlying biological processes specific to B cells in their differentiation and maturation. And therefore, in theory, this should perform very well in comparison to more universal but less specific methods. So in future, we will aim to expand the use of this method to other types of transcriptomic data, such as spatial transcriptomics, so we can have a look at further other subtle information in instructing the dynamics of state transitions. So the method is already available as a repository, and if you want to learn more, please go to this preprint on the bioRxiv to learn more about the implementation and details of the methods. Thank you.